educational institutions to help them become world-class teaching and research institutions. IIT Madras is proud to have been selected as one of the Select Institute of Eminence. A total of 68 research initiatives belonging to 21 identified clusters are presently underway at IIT Madras. As a part of these initiatives, the IRIS webinar series aims to showcase the innovative research that is being generated to various stakeholders like students, researchers, industrialists, and policymakers. So this is Rudra on the behalf of the Office of Global Engagement. It's my pleasure to welcome you all for the 49th webinar from the IRIS webinar series. So from the medical and biotechnology cluster, this research initiative presenting today is molecular medicine led by Professor Nitish Mahapatra. This research initiative aims to identify new molecules and therapeutic agents for treating chronic diseases like cancer, cardiovascular disease, and neurodegenerative diseases. So the speaker for today's program is Professor Nitish. So he has published more than 60 research articles in reputed international journals, including Journal of Clinical Investigation, Journal of Biological Chemistry, Cardiovascular Research, and Hypertension. So Professor Nitish's laboratory investigates pathogenesis of cardiovascular and metabolic disease, including hypertension and type 2 diabetes. He has received a number of awards and honors for his scientific contribution, including Fulbright Nehru Academic and Professional Excellence Fellowship, Torrent Research Award from the International Society for Heart Research, and JSPS, which is Japan Society for the Promotion of Science, in Invitational Fellowship. He is an elected fellowship of the National Academy of Sciences India and America Heart Association. So thank you for being with us today, Professor. We are excited to have you, that you could be a part of the session. And our moderator for today's program is Dr. John Osborne. Dr. John is Marvin Bachner Endowed Chair in Cardiovascular Physiology at the University of Minnesota. His laboratory is interested in understanding of the role of the central nervous system in the long-term regulation of arterial pressure. The primary research goal is to understand the pathogenesis of hypertension and provide translational platform for development and novel therapies. And he has published more than 150 research articles in well-renowned journals like the Journal of American College of Cardiology, Circulation Research, and Hypertension. So before we start, a note to the participant, please use the Q&A box to enter your questions and like or upvote the question that may interest you so that the moderator can prioritize them. Over to you, Professor John.
Was this turned over to me just now?
Yeah, I mean, you can just welcome Dr. Nitish to start the session.
Very good. Dr. Nitish, you want to take it over?
Sure. Thank you. Thank you so much. So thank you, everyone, for attending this event today. And so let me share the screen. So is my screen visible?
Yes, sir.
Okay, good. So, good evening, everyone. So today we are going to talk about a new center that we are establishing here over in IIT Madras. So the center is called Center of Excellence in Molecular Medicine. So let me, so I'll be kind of briefing you about the objective of the centers and the participants of the center. And as you have heard, the moderator for this today's session is Professor John Osborne from the University of Minnesota. We are truly thankful that he could make time to attend this event. So the investigators for this particular initiative includes myself. So I work in the area of cardiovascular and metabolic diseases. So particularly my lab is interested to discover the molecular and genetic basis of cardiovascular and metabolic diseases, including high blood pressure, type 2 diabetes, coronary artery disease, and related disorders. And we have also Dr. Suresh Royala, who works in the area of cancer biology. So his lab is looking into various novel therapeutic aspects of cancer biology. And then we have Dr. Mozulika Dixit. Her lab is interested in the area of vascular biology, endothelial dysfunction, and atherosclerosis. So their lab uses various tools towards understanding the mechanisms of endothelial dysfunction and type 2 diabetes, how this could be related, and in order to develop newer kind of therapeutics for atherosclerosis and related disorders. Then we have another investigator, Dr. Amal Kantibera. He is an expert in the electrophysiology. So his lab works on ion channels and signaling, and his lab is primarily focusing on various neurodegenerative diseases, including stroke, how to develop newer kind of molecules which could be used for treating stroke. And then we have another investigator, Dr. Nidita. She works in the area of cardiovascular biology, neurophysiology, and metabolic syndrome. So primarily, she is interested in finding out newer mechanisms to understand hypertension and related cardiovascular disease conditions, with a special role in the special role the neuronal aspects play in the development of cardiovascular diseases. That is the area of our expertise. So we five faculties from the IIT Madras are interested in setting up this center called Center for Molecular Medicine, and the overall objectives of our center is to identify novel drug targets and active principal ingredients, APIs, such as flavonoids or poly-ADP ribose-polymerase or PARP inhibitors, and certain microRNA based therapeutics which could target the common diseases like cancer, atherosclerosis, hypertension, and stroke. As all of you are aware that each of these disease conditions are quite prevalent, not only in India, but in most countries. So, but in spite of tremendous amount of research over the past several decades, still each of these disease conditions are so much prevalent. So we need to actually put our efforts in a more concerted manner to come up with newer kind of therapeutics. So that is the main objective of this center. So we have kind of four specific objectives. One objective is to assess the role of microRNAs. MicroRNAs are small non-coding RNAs which are emerging as very important regulators for gene expression, and those could be very important in various disease conditions. So here we aim to assess the role of certain microRNAs which could be useful in the treatment of cardiovascular disease and cancer. So I will talk about each of these objectives in the next few slides. So let me first introduce you to the specific objectives. Apart from the microRNA-based therapeutics, we are also interested in evaluating the role of phytochemicals as anti-atherosclerotic agents. So herein we would like to see whether various phytochemicals can act as effective treatment for atherosclerosis. Another objective of this project is to identify ion channels as potential drug targets for treating stroke. And last but not the least, objective of our center is to elucidate the role of poly-ADP ribose-polymerase. In brief, this is called PARP, whether inhibiting PARP could help in treating heart failure and related conditions. So I'll tell you about each of these objectives in little bit details in the next few slides. So coming to the role of microRNAs in cardiovascular diseases and cancer, microRNA therapeutics is emerging as next generation strategy to treat a wide array of human diseases, including cardiovascular disease and cancer. That is mainly because microRNAs are small molecules, and then this can be synthesized in the laboratories, and each microRNA could actually regulate several genes, as I will be showing you in next couple of slides. In fact, there are several clinical trials are going on to develop microRNA-based therapeutics in order to see if those can be used as effective therapeutic agents. So indeed, there are several potential molecules in the pipeline of microRNA therapeutics. So in our laboratory, recently we have shown that microRNA 27A could abrogate hyperlipidemia in the ApoE knockout mice. ApoE knockout mice is a commonly used model for atherosclerosis, where the animals would develop atherosclerotic plaques, and their cholesterol levels become elevated. So what we have shown here recently that treating these animals with microRNA 27A mimic leads to diminished cholesterol level in the plasma, diminished LDL cholesterol level, as well as the triglyceride levels in these animals. So when we check the different enzymes for the liver function and kidney function like ALT, AST, and creatine kinase, we see that their levels were unchanged in the microRNA 27 treated group as compared to the control group, suggesting the potential of microRNA 27A as a potential therapeutics in this disease condition. So looking at the mechanism of this ApoE knockout mice, how these cholesterol levels could be diminished, we found that microRNA 27A actually targets multiple genes in the cholesterol biosynthesis pathway. So here, each of these red color symbols are indicating the different enzymes present in the cholesterol biosynthesis pathway, which are targeted by microRNA 27A. And we, in fact, could validate that several of these enzymes are indeed targeted by microRNA 27A, which is probably the mechanism by which the cholesterol level could be diminished upon treatment of the ApoE animals with the microRNA 27A mimic. Indeed, the microRNA 27A augmentation in the ApoE knockout mice could rescue atherosclerosis. So what we did here is essentially we injected microRNA 27A mimic using some liposome-based formulation, injecting this liposome-based formulation into the tail vein of these ApoE knockout animals, which are fed with high-fat diet for a period of time. And then we found that this could lead to the lower level of the plasma total, as well as LDL cholesterol, improved cardiac function, as well as reduced plaque formation in these animals. So this has very interesting these interesting results are kind of suggesting that this could be useful to study further as potential therapeutics for atherosclerosis. Okay, next, microRNA 493 was found to inhibit the tumor growth in the nude mouse models. So this was carried out in Professor Suresh Arala's lab, where they have provided convincing evidence that if you treat the nude mouse models, the tumor growth could be significantly reduced by expression of microRNA 493. So there are, as you can see from this slide, the control animals had the tumor growth, and this could be the tumor sizes are effectively reduced upon treatment with microRNA 493. And the mechanism involves various important molecules in the uh, in the uh, um, uh, in the tumor suppression pathways, and one can see that IGFR, IRS1, MAP kinase, snail, these are key molecules in the cancer regulation, so they are all kind of suppressed upon treatment with the microRNA 493. So this study from Professor Rawla's lab clearly provided evidence for the snail modulated microRNA 493 inhibition of the IGF-1 receptor pathway and blocking of the tumor regenesis. So this study clearly showed that MID493 is a tumor suppressor microRNA, and it can target IGFR, IRS1, MAP kinase pathways. And again, this microRNA 493 expression is regulated by snail, and the microRNA 493 regulation of snail expression is mediated by IGFR1. Indeed, the study showed that there is an existence of a feedback loop between the IGFR snail and MID493, and also they showed that role of nicotine in tumorigenesis affecting the above feedback loop. So, as you know, that nicotine is a molecule of interest for various cancer. So here, the effect of microRNA 493 is indeed very interesting. So then, this another project where we are looking at the role of potential phytochemicals in as effective atherosclerotic agents, anti-atherosclerotic agents. So here, this study was carried out in Professor Madhulika Dixit's lab, where they have provided clear evidence that the root extract from certain plants, which contains the glycosides, which could show tremendous effect on the atherosclerosis. As you can see from various experiments done over here, the control elements, if you compare between the control and the extract treated animals, as well as they use the ST rat, which are streptozotocin rats, a very useful model for diabetes and atherosclerosis. So it shows that the atherosclerosis plaques were significantly reduced upon treatment with this glycoside-rich root extracts. So many studies have been published from Professor Dixit's labs in this area. So they have a study approach where utilize the endothelial cells, which are isolated from the umbilical cords, thereby using the human umbilical vein endothelial cells, and also endothelial progenitor cells, smooth muscle cells, using various cell culture model, patient samples, as well as mice models. It is possible to study these anti-atherosclerotic effects of various phytochemicals. So these are being carried out in Dr. Madhulika Dixit's lab. Then Dr. Amal's lab, Professor Amal Bera's lab, is working on ion channel targets for different kind of ion channels, which could be used as a potential target for treatment of stroke. So they have studied several ion channels, particularly the target ion channels that are being investigated in Dr. Amal's lab are acid sensing ion channels, ASIC, and volume regulator ion channel, or VRAC, which are important target molecules for stroke. So in animal models, it has been shown that blocking of the ASIC and the VRAC reduces stroke-induced brain damage. And in fact, there is no therapeutic molecule available for stroke patients as of now. So these studies are very useful in this direction. So because the ASIC is implicated in the expression of the peripheral and central nervous system, overactivation of low extracellular pH during stroke, and it also causes increases in calcium efflux, influx leading to the neuronal death. In case of VRAC, this is activated by cell volume changes and it releases glutamate and causes neurotoxicity during stroke. So these are very important molecules that need to be targeted. And studies from his lab has shown that like quercetin, which is a polyphenol, which could be a very effective blocker of the acid channel. So they have provided convincing evidence that the ASICs or ASIC channel is blocked by quercetin. The ASIC was able to block quercetin was able to block the ASIC-mediated current over here, as well as the calcium. So you can see that in a dose-dependent manner, treatment of quercetin is leading to the blockade of the ASIC-mediated current. And it has also been shown that the quercetin molecules are binding directly with the ASIC. So these are very important studies in this direction. So the workflow for identifying therapeutic molecules for targeting ASIC and VRAC involves in silico screening of the flavonoid library and FDA-approved drug library for ASIC and VRAC blockers. And that would be followed by high-throughput screening of selected molecules using cell-based assay system, and that would be confirmed by PASCLAM assays and using primary neuronal culture, which will be followed by hypoxia reperfusion model using primary neuronal culture. And also his lab has a zebrafish stroke model, so wherein the animals would be treated with these various molecules, flavonoids, and then their effect will be tested. So this is a very exciting area of research. Then another project where Dr. Nirita is working on is on the role of PARP molecules, which are poly-ADP ribose-polymerases, how they can be used for treatment of heart failure. So poly-ADP ribose-polymerase or PARPs are multi-domain post-translational modification enzymes. So these play a vital role in aging, cell cycle regulation, metabolic regulation, and inflammation. So each of these pathways are very relevant for cardiovascular and cancer pathways. So PARP inhibitors have been identified as anti-cancer agents. So here the idea of Dr. Nirita's laboratory is to test the role of different PARPs in the pathogenesis of hypertension and hypertrophy, which is a kind of precursor for heart attack. So these are not clearly understood. So our lab is pursuing experiments in this area. The aim is to identify whether PARPs can alter the signaling pathways and mediate hypertension development, as well as cause hypertrophy in the relevant animal models. So she is an expert in various animal models of hypertension, where she has extensively published in highly reputed journals, including renal denervation model and angiotensin II salt hypertension models, as well as various transgenic models that she had worked on. So these models are going to be used for finding out if PARP inhibitors could act as effective molecules for treatment of cardiometabolic disease states. So we are fortunate that we have several international collaborators. They are from various universities, various reputed universities from different countries, including Professor John Osborne, who is present here today as the moderator of this event. And we have various investigators from numerous places around the world, and they are going to help us in carrying out these projected studies. So overall, our anticipated outcomes of this center is that we'll be able to come up with various herbal and Ayurvedic formulations or flavonoids for neuroprotection and PARP inhibitors for cardiovascular disease and novel microRNA-based therapeutics for treatment of cancer and cardiovascular diseases. So these are different disease states that we are looking at in order to, each of these are important. So we need to study each of them we have taken a concerted approach in order to study these. So I would like to conclude my presentation over here, and any additional information or updates can be obtained from our website that is mentioned over here. So now I stop, and I would request Dr. Osborne to take over and ask any relevant questions that he may have.
Thank you, Dr. Nitish. That was great. Can you all hear me okay?
Thank you. Yeah, you are audible.
Great. So first of all, I want to say it's an honor to be included as one of the international collaborators for this project. And to the students in the audience and everyone else as well. I mean, linking the molecular mechanisms of these diseases, cardiovascular, cancer, is a huge task, but it's extremely important. I mean, we're desperate for therapies for cardiovascular diseases. Hypertension, heart failure are huge. There are no new drugs in the pipeline right now for these diseases. Cancer as well. So, you know, this particular area that's emerging in terms of microRNAs and some of the work you've heard here is critical, not just, as you said, in India, but everywhere. So for the students, this is an extremely exciting effort to get involved in these labs. So that's an exciting time for you all. Now, Dr. Nitish, one thing I want to ask about, I mean, so the approach here is looking at a certain level of biology. Eventually, we need to translate this into therapies, correct? So can you discuss a little bit the idea of the translational aspect of this to go from these models into the clinic? So there's a lot of work to be done here, a lot of basic science to be done, but the translation is a tough part. Can you comment about that a bit?
Yeah, so yeah, it's as you correctly said that these are all kind of long-term projects. We are basically initiating at the level of finding the signaling pathways and the potential molecules which might be involved. And so once we have the preclinical studies done, then once the target molecules which are showing to be most promise to take to the next step, then we will collaborate with other laboratories as well as some companies who are working in this area, so that we can go to the next step, which is like going to the development of these potential therapeutics. So it is indeed a long-term process, but we are hoping that in the long term we'll be able to come up with some molecules which will be useful for treatment of the different disease conditions.
And of course, the challenge, but also the beauty of this approach is many of these signaling pathways can be involved in different disease states and in different organs, right? So that's
Yes.
a challenge, but it's also gives you an opportunity to, when you integrate your work together as this team, that you can find common pathways that are involved in more than one condition. So that's a real strength, I see.
Yeah.
Well, there are some questions in the Q&A here that maybe we could ask.
Yeah, we can take some of the questions probably from there, and any of us, like we have our in the panel, we have all the investigators and yourself. Anyone can answer any of these questions. So could you propose some of the questions? Then
sure, let me read one of them here. PARP is usually implicated in cancers since it's one of the DNA repair mechanisms adapted. Is DNA repair pathway abrogated on heart failure too?
Yeah, so I think maybe Ninita can answer that question because she is directly working in that area. Ninita, will you want to be able to take that question?
Sure.
Yeah.
Hope I'm audible.
Yes.
Yep.
We have some preliminary evidence which supports that in mouse models, parts are involved in the cardiomyopathy. So we have some preliminary data which confirms that PARP's inhibition may be involved in the development of cardiovascular diseases. The role in hypertension has yet to be explored, but we do have some evidence.
Let me follow up on that. One of the questions I had is sometimes it might be advantageous to do organ-specific targeting of some of these pathways, right? So if we're thinking about heart failure, maybe the heart, is that, what is the possibility for that in terms of targeted interventions? That'd be for anybody here, Nanitha.
So the research is, yeah. So as far as hearts and heart failure is concerned, it is very preliminary to say that which organ should we be targeting. Yes, cardiac tissue may be the one, but are the nerves talking to the heart involved in it? We have to do further research on it. Maybe others can chime in.
Yeah, not only for PARP, probably for other molecules as well, as Dr. Osborne correctly pointed out that some tissue-specific models probably would be useful. So we have to develop some models, or we can do that in collaboration with our investigators in the various places, that some tissue-specific knock-in or knockout models should be useful to come up with more valuable answers to those questions.
Mm-hmm. Okay, here's another question. So how was the target genes of MIR27A identified, and what do they do with those genes so that the cholesterol biosynthetic pathway becomes hyperactive?
Okay, so yeah, basically there are different computational programs now available by which one can find out what are the target genes for particular microRNAs. So here we use some of the standard tools for microRNA prediction of various, like there are two ways to do it. If you have some target gene, you can find out which microRNAs target those. And on the other hand, if you have a microRNA of interest, you can find out which genes are targeted by that particular microRNA. So there are standard tools now available. One can carry out those analyses to identify the potential target genes, but that is actually not very conclusive. That would give you a huge list of target genes, because each microRNA can target hundreds or thousands of genes. So in order to identify which of them are truly interacting, which of the target genes are truly interacting with the microRNA of interest, one needs to carry out several experiments. One such experiment is called like 3' UTR reporter assays, by which 3' UTR of different genes are primary targets of microRNAs. So one has to do individual 3' UTR of different genes cloned with a reporter, and then try to see if they are responding to this microRNA overexpression, or using antagomics, which is a group of reagents which are used as an antagonist to the microRNAs. And then one can find out if certain genes are truly targeted by the microRNAs. So here what we did the same approach. So if you have time, just go through our molecular cell biology paper, and you will see the exact protocols that we have used.
Mm-hmm. Thank you. Let me see here. What is the role of cholesterol in heart failure? I'll start with that, but I'll let Nanitha or anyone else jump in too. I mean, so my lab studies hypertension, but we know high blood pressure and metabolic diseases go hand in hand, right? So many people with high blood pressure have problems with glucose metabolism or diabetes. They have problems with lipid metabolism, and those can result in heart failure, either as a result of high blood pressure, chronic high blood pressure effects on the heart, or direct effects on the heart. So it's complicated interaction. We think about metabolism and cardiovascular disease. They're almost always linked together. And getting to the role of this center, trying to identify potentially common pathways that are affected is important. So the short answer is we don't really know exactly how they're connected. They can be either indirectly connected through changes in the cardiovascular system that affect the heart, or through some of these signaling pathways. I don't know if anyone else in the center can address that in terms of the link between cholesterol and heart failure.
Yeah, probably elevated level of cholesterol is almost always connected with atherosclerosis, and that may lead to heart failure, but not always. Actually, particularly in India, we are seeing a lot of people who are getting heart failure at an early age. So they are like less than 40 years, or sometimes even at early age. So when I talk to my clinical collaborators, they say that these people who are getting the heart attack at such an early age, it is not due to cholesterol deposition in their arteries. It is rather due to kind of rupture of the arteries, sudden rupture of the arteries, and it is not due to high cholesterol. So this is mostly due to some sort of endothelial dysfunction, that they are endothelial layers are somehow brittle, so that that gets in response to sudden shock or sudden stress, these people are getting the heart attack or heart failure. So which is somewhat different from the usual heart attack, usual heart failure cases where elevated cholesterol should lead to the blockade of the arteries, and then that might lead to the like the blockade of the supply of blood to a particular portion of the heart, leading to the myocardial infarction. So it's not always that elevated cholesterol is directly connected with the heart failure, but in a large number of patients, particularly the elderly heart failure patients, elevated cholesterol is indeed a major risk factor for heart failure.
Thank you. I want to ask, there's a question in here that I think is really important to address. Are there side effects of miRNA as therapeutics? And this is the issue with any therapy we develop. Does it work? Is it effective? But are there side effects? And oftentimes the side effects derail it as an effective therapeutic. So can you address that, Dr. Nitish?
Yeah, that's a very good question. Actually, there are several clinical trials that are going on. So I have cited a couple of these papers in the presentation. So here I see that several of these in the clinical trial phase 2, and one of them has entered phase 3. So they are clearly people are evaluating the side effects. So it would be too early to say that whether the microRNA that we are going to test or taking further for clinical trials, whether they will have side effects or not, is much earlier to say anything in that regard. But obviously, Dr. Osman clearly mentioned that any molecule before going to the clinic needs to go through the different phases of the clinical trials, and only if the side effects are kind of minimal and if the benefits kind of outweigh the risk, then only the microRNA-based therapeutics can go to the clinics. But we are still at the early stage.
Yeah, and that's a challenge with these signaling pathways being in lots of tissues, and another reason to do tissue-specific targeting if possible, which is a big task, but that's always the way we're thinking, right?
Right.
Yeah. Another question here, and I know we've just got a few minutes left. How are miRNA specific to cancer cells? That kind of gets to what we were just talking about a bit.
Okay, so I'm going to answer this. So like microRNAs may function both as either as oncogenes or tumor suppressors under certain conditions. So depending on the dysregulated microRNAs, which have been shown to affect several of the hallmarks of cancer cells, which means that if a microRNA is identified specifically and it targets the 3' UTR, I'm sure it will specifically target the cancer cells depending on the target.
Thank you. Anyone else want to add anything to that? Okay, let me ask one. There's a couple of questions that have come up related to the role of microRNAs in neurodegenerative disorders, which we haven't talked about that much. Is there a role in, so for example, Parkinson's, Alzheimer's, those kinds of things? I don't have any knowledge of that myself, but anybody?
The role of therapy of neurodegeneration.
I can answer.
Okay.
Yes, it does have several roles, but not really studied well. We know that it is associated with several neurodegenerative diseases, but it is not really or fully understood yet. But yes, it does have role.
I'm sure the answer is going to be yes, as you look more and more into
that's what I'm saying. But no detailed experiments need to be done. We don't have detailed understanding.
Right. Let's see here. There's a few more rolling in. Getting back to, we were talking about, where was this? We were talking about heart disease, and actually this has disappeared, sorry. We just have a couple of minutes left, I believe. Is that correct?
Yeah.
I mean, Nitish, do you want to make any kind of general comments related to this? And then there's some of these questions I think we can get to after the session would be the idea.
Yeah, sure. So, yeah, basically the center that we are trying to build would be interested in setting up collaborations with the different labs, and we will be also looking at research scholars and postdoctoral fellows who are interested in this area, so that they can participate and take this study further. And overall, this is a long-term mission for our department, and we are kind of very optimistic that we have a good array of collaborators, so who are expert in the different areas, including your lab. So we will be approaching each of them for their expertise, and we plan to conduct various meetings and workshops. Once the pandemic is kind of probably settled, then we will have an opportunity to directly interact, and maybe some student exchange visits would be possible. So these are very important to take the work to the next level. So yeah, I would like to just share that we are basically initiating this center. We would need help from each of our collaborators, and we are open to any kind of ideas and any sort of support that may help us to grow.
Yeah, I will add to that just in closing. I think it's really exciting, and the way this will be successful is the connection with collaborators and the students and postdocs you bring in to work on these projects, to link these labs together, you know, across the world, basically, right? So, from my own experience trying to work with large collaborative groups, it's really facilitated by the PhD students and the postdocs, and getting together and exchanging ideas, because it is a big task, but it's really exciting to do that. And then to bring in the translational approach too requires that kind of effort.
Yeah.
Any other concluding comments here? I think we've run out of time. Is it
Any
possible
answer
to answer questions that we didn't get to after the session is over?
Yeah, actually, like some of the questions probably can be answered. If you have another meeting or something, you can also leave. I think we can continue answering some of the remaining questions. And basically, the main part of this event is over. And we can, if some of our panelists are interested in taking up any of the questions, they can take up and continue the discussion for some more time. But anyhow, I think I would like to thank you so much for making time and joining us today.
My pleasure.
Thank you so much.
Yeah, we'll be definitely in touch, and then we'll seek your help at different times.
I'm looking forward to that very much. I've been to India 3 times. I'm looking to coming another time.
Sure,
So that would be great.
Most welcome.
Yeah.
Thank you, Dr. John. It's our pleasure to have you here. And probably if Professor Nitish and his team wishes to take the question, some more questions, maybe you can still continue the session.
Okay.
Okay, that sounds great. Thank you, everybody.
Okay. Yeah.
Professor John.
Bye. Take care.
Have a good day. Yes, sir. Over to you, sir. Maybe if you can take up the questions, some more questions, probably we can.
I think Nitish, most of the questions are repeated only. So.
Okay, so is there any question that you think could be?
I don't see any important questions we should
Yeah.
Yeah, in case there is no relevant question, probably we will conclude.
We'll conclude probably. Yeah.
Okay. Okay, that's fine then. So I think there is no relevant question. So I think some of the questions are like non-technical. Someone says, I did my bachelor's in biology.
Yeah.
So I think we can't answer those.
I think yeah, we can end the session.
Yeah. So what about Rudra? Did you or anyone from the GE office answer those questions, like general questions from this?
Like we will take care. We will take care of it. Please don't worry about it.
Okay.
We'll take care of it for the general question. And also, I did see a question from Ankita Yadav, like, sir, how can I join your lab for internship? But I'm not sure, like, if this is a possible answer if you can give. But still, there are a lot of requests from the students and the researching community as well to join IIT Madras, maybe either in the like an internship or to work with us. So probably if we can have.
So we can just say like the procedure by which they can get into this, the general procedure for joining.
Summer interns.
Summer interns will be they can apply when like this year we didn't have the IIT Madras summer internship because of the pandemic, but next year probably we'll have it. So at that time they can apply online, and then some of them will be selected based on their credentials. That's for the internship. And regarding PhD or postdoctoral applications, they can directly apply at the department websites at the different times when the advertisements are made, so that we can consider their applications.
Right.
Anything else? Madhu, do you want to add anything? You didn't say anything.
No, I think you almost cover everything, Nitish. Thank you. I don't have anything much to add.
Okay, in that case, probably we'll close the session.
Sure, sir.
Okay.
Thank you, Dr. Nitish. Probably we can just close the session. Thank you, audience, for your participation.